Hello, good morning. It's 8:15 and we have a tight schedule. We have only 1 hour before the next study group wants to come in. So let's get started. Feel free to get coffee and croissants during the meeting as well. It was very expensive, so it must be excellent coffee. Yeah. So this is our agenda. As usual, we want to talk about the current topics and issues in our field, in the studies group specifically, but also in the research field that the study group is concerned about. We'll talk about the demographics as we have to, and a bit of what about the things that we did in the past and we intend to do in the future. We'll talk about the new governance committee, and then also there's some content at the end with the trainee awards of the poster presentations. Okay, so as a reminder, we have some communication channels. Specifically, there is this listserv, brain_function@ismrm.org. It's quite quiet, and it doesn't have the best reputation because it's heavily conservatively moderated. However, the good news is that the moderation is opening up a bit. So now we get approval that we are allowed to blast things at this listserv that is relevant for us, but not directly organized by the ISMRM office. Yay. Also, they will roll out this new way of doing communications with this higher logics. We don't really know what this is yet, but we will learn soon. It's supposed to help the committee better communicate with the members and also to other study groups. There's also this Discord forum, which is pretty dead, I think, but you can use it, and I think it also sends reminders to some people. It's not as heavily moderated. It just takes a while before the reminders are sent out. So if you want to advertise your conference or your event or whatever, send it early. We are still struggling as the committee of the Brain Function Study Group to get all this amazing content that ISMRM has on educational material for fMRI out there to our members, specifically without the paywall. The ISMRM Academy, or MR Academy on YouTube, has a bit, but the fMRI lectures is really underrepresented. There may be 2 or 3. And the stuff that we did ourselves, like having YouTube playlists, is still offline as we were threatened by ISMRM lawyers. Then we have, outside of our ISMRM environment, there are still communication channels and outlets that I think I want to advertise because it's ISMRM and for us important, specifically for fMRI method developer. There's the transmitter feature with the future of fMRI. Also in OHBM, they have this podcast, which is concerned about OHBM topics, the neurosalience. However, there's a lot of overlap specifically with this study group, and there have been specific episodes where we talk about OHBM and ISMRM and basically the lack of crosstalk there, and we should work on that. And who but us, our study group, would be the best partner to make the connection to OHBM, and maybe in the future there might be joint workshops, as Patricia will tell you. Also, there's this new edition of the Encyclopedia of the Human Brain with exciting fMRI method developers chapters as well. Check it out. So we had this workshop organized 1.5 months ago in Annapolis. This was a joint organization between the Brain Function and the Ultra High Field Study Group, which was a perfect match. It was a nice workshop with more than 200 people, and the content and the recordings is all online now. It's behind the paywall, but if you don't have access, let me know. We find a solution. The new trends in the field that we should be aware of as a study group and in kind of shaping us and see where we are going are kind of evolving a bit as opposed to the last years. For example, still a hot topic is CSF imaging and like the neural fluids, where from the methods perspective, a lot of fMRI method developer have kind of moved on to this direction. So we need to keep close watch and be in good communication with those study groups as well. Something that the global fMRI field is moving on, and we are kind of a bit behind in the ISMRM community is these global standards on how you should present your data, specifically how you should present your activation maps. So there aren't these community standards with almost all of the major software providers suggesting that you should present your activation maps as these semi-transparent thresholding. So we need to pay attention to that because as ISMRM folks, at some point we will need to get past peer review from those non-ISMRM folks that impose those standards as well. With respect to fMRI being represented in other fields, we have new field strength. The 5 Tesla fMRI is being established more and more. For example, 5 Tesla fMRI, layer fMRI, also with Bold and Vaso, and also the very first fMRI in vivo human fMRI results at 11.7 Tesla acquired, with exquisite EPI and also the first QA-style kind of connectivity and localizer activation maps in humans at 11.7 Tesla. yay, fMRI. Also one method that is getting a lot of attention within ISMRM, but also beyond, that we should keep close watch, and I want to give it another shout out as well, is this tidy method, which gives amazing results. Check it out on Thursday. As for controversies that we need to keep track of in our field, specifically the different fundings environment now, for the purpose of fMRI methods developers, already last year, kind of the BRAIN initiative has kind of declined and reduced more and more and more. There are still a few fMRI grants, method developer grants funded by that, but it's going to be less and less for sure. Also, the topics that are fundable are kind of being shrunk. For example, if our friends in the study group doing preclinical work are specifically targeted. So it's kind of very worrisome if maybe this room next year will be half of the size if we don't have funding. And there are slow kind of initiatives out there to talk about the threat on science, globally on science, but it's not really clear what this means for us, specifically fMRI, as we do also preclinical animal stuff. The very sad news of the year is that Rosa Sanchez Panjuelo has passed away. She was an active member of this community, and we miss her a lot. As for demographics, we are still increasing in member numbers. We had a bit of a dip during COVID, but last year we increased again. Now this year we increased even further. Geographically, it's basically US and the rest, like by far. There's a bit of China and Canada, but basically an order of magnitude more than US than the rest. So any kind of the funding changes in US are basically relevant for this study group very much. We still have a gender bias. It's less, but it's still there. We need to work on that. And we have a quite healthy trainee representation still, as always. And with this, maybe I can hand it over to Daniel, who will talk about some stuff that we did in the last year, specifically the virtual workshops. Thanks.
Hi, everyone. I just wanted to tell you about the past virtual workshops that we organized. One in March 25th last year, where I and Camille organized this workshop, and we had Gary Glover, Maria Guidi, and Samuel Bianchi giving talks on the HRF. And we also had a workshop later last year on multimodal imaging of functional brain networks with pharmacological fMRI, co-organized by Tony and Cesar, with speakers Andrea Lupi and Ottavia Di Pasquale. So I'd like, on behalf of the study group, to thank the speakers. And I would like to mention that it's very easy to organize a virtual workshop. All we need to do is fill up a form. So if anyone has suggestions, we just send emails, we can get the speakers, and we can organize them. It's super easy. So yeah, with that, I'll give it to Patricia.
Thank you, Daniel. Good morning, everyone. Good to see you all here. So my first role as the chair for the next year is actually to say thank you to two members who are leaving the committee now. One of them is Daniel. Thank you very much for being a great trainee representative for the past 2 years. And the other one, unfortunately, cannot be here, but we also thank him. Camille Uludag was the chair, and he was on the chair track for 4 years between 2021 and 2025, so a big applause to him, too. And now I'd like to announce the new committee members who unfortunately are not here in Honolulu with us. So I just thought I'd briefly introduce them to you. So we have Saskia Bowman as the new secretary. She's a research fellow at the University of Queensland in Australia. She develops new contrast mechanisms for fMRI and investigates the effects of vasculature on the fMRI signal. Probably you all know her. So we're very happy to have her on board. Then we have Nikos Piovolos as our new trainee representative, joining Tony. He's a postdoctoral researcher at the University of Oxford, and he's an MRI physicist and neuroscientist interested in characterizing both brain structure and function quantitatively in vivo and reproducibly. And finally, we have Rebecca Glarin. She's actually not new in the committee. She's been our ISMRT representative for the past years already, and she's agreed to continue in this role. If you are aware of any radiographer interested in this, we are having some difficulties, so Rebecca was very generous in agreeing to continue. She's also in Australia at the University of Melbourne, and she's doing her PhD as a radiographer at the investigation of the sympathetic drive by combining the use of microneurography with ultra-high field fMRI. So now this is the new committee. So Renzo, myself, Cesar Caballero Gaudes as vice chair, and yeah, Tony, you'll hear from her in a minute. Okay, so this is time for me to actually pass the word on to Tony for the trainee awards, the most exciting part of the meeting.
Okay, hello everyone. Now we're coming to the thing that we all waited for today. We invited 4 trainee members of our community to give a talk today, and we can already now actually applaud to them because they already made it to this selection. So this is already really great. So how this is going to work is that they have 5 minutes to present their work to us. And please keep them in mind, because afterwards we're going to have a little audience vote for them, and you can vote for your favorite presentation, and they can win some prizes. And with that, I would already like to invite the first speaker on stage. It's Nadira del Rosario, and she's going to present fMRI using 1D line scan and 2D imaging, are also often equally sensitive, comparing SNR efficiency in theory and practice.
Thanks so much for the introduction. I have nothing to disclose. So conventional laminar fMRI is limited by imaging resolution, where you usually need to go to smaller and smaller voxels in order to actually get a better idea of what's happening at critical layers. And most importantly, hemodynamics are often regulated at this much finer spatial scale. So, it seems like the solution is super easy, just have smaller voxels, right? But of course, in reality, it's not that easy. But one way that you could do it is by using 1D laminar line scan fMRI for laminar fMRI. And line scan is actually a classic imaging method, and it's recently seen increased application in high-resolution anatomical and functional imaging for critical layers. And this is because it actually gives us really, really high spatial resolution along the line. Oh, okay. Imagine there's a figure there, and it's showing you a brain with, it's just not coming up, with a line across the motor cortex. And then imagine that we're zooming in there, and we're seeing these cortical layers. Now, we already know that critical layers are actually pretty small, way smaller than 1 millimeter voxels would actually allow us to see. Line scan gives us this very, very fine resolution of sub 0.25 millimeters. And when you align these to the cortical layers, it actually approximates the true biological resolution of these layers. And that's a big benefit of line scan. So if it's so good and it gives us all these benefits, what's keeping people from using it? Well, oh, okay. Well, it's because you saw it. It's because despite these benefits, there are still some concerns that remain about line scan's viability for mesoscale imaging, but these have mostly been addressed. One of them is movement, another one is line prescription, and another is coverage on the brain. Today, I want to talk about SNR, which is the remaining thing that has not yet been addressed. And considering theory and practice to demonstrate that the SNR efficiency of 1D line scan 2D imaging are comparable. We'll test the hypothesis that 1D and 2D acquisitions are often equally sensitive, and this is a concept that was previously shown for 2D and 3D imaging when using Ernst and when these are matched for spatial and temporal resolution. So when we talk about matching, this is the 3D volume and this is a 2D slice. And you can actually use the same volume TR for the acquisitions, and then you can use this for everything. We can do something similar to match 2D and 1D acquisitions in k-space lines using the same temporal sampling interval. And then we do this for all of our acquisitions. And so we hypothesize that because this increase 1D signal recovery that you're going to get, you actually will have similar SNR efficiency when you compare it to 2D acquisitions. But there's still additional factors that can impact SNR. The way we're addressing these is using anisotropic 2D voxel flash and comparing it to 1D line scan with dual saturation and outer volume suppression. And one of the things that we have to consider is that the imperfect saturation bands can affect the quality of the slice or line selection profile. Of course, the imperfect saturation bands themselves can affect our signal, and but we still benefit from line scan being a single shot technique. So here I'm showing you what the anisotropic 2D voxel flash would be like, and here is the 1D line scan sequence that we will be using. This is how you would get the saturation bands once they're on, and then at the end you get this very nice crisp 3 millimeter line. So I'm going to address the imperfect saturation bands because these can actually cause changes in the signal along the line. So if we place these like right around the edge of our phantom, very similar to what we would do in an experiment, and then we look down the very center of our phantom, and then we turn both of these saturation bands on, you can actually see again this very crisp line that I was mentioning, but you can see that it's not entirely perfect. There's still a little bit of signal outside. When we look at what that signal looks like right down the line, you can see that there's something that looks like a little bit of an artifact. That's the light blue line. That can potentially be due to aliasing, but we're still trying to understand what that might be. Finally, we then compared sensitivity, first in an agar phantom between 2D and 1D, and then that's crazy. Anyway, look at the bars. What matters here is that when you compare both of these, their SNR sensitivities are actually, the efficiencies are actually quite similar. And even though you can still see a little bit of that aliasing going on that we're still trying to understand, but it's probably due to signal getting wrapped into the line. And then we did the same thing in a human scan with these equivalent 2D and 1D acquisitions, and we see something very similar. So in summary, it seems that 1D and 2D fMRI appear to be also often equally sensitive, with the phantom and human data suggesting the efficiencies between 1D and 2D acquisitions being quite similar. However, we can often see these higher signal levels due to artifacts that can contribute to apparent higher SNR and line scan acquisitions, potentially due to the aliasing. And we still need to do a little bit more work to determine the impact of the physiology on TSNR when we're using these methods. And if you want to know more details, please do check out my poster. It's up there. You can see it. Thank you so much for listening.
Thank you so much. Then I would like to invite Janne Kananen on stage. He's going to present reduced relative harmonic power of diastolic cardiovascular impulses in Alzheimer's disease.
Hi all, and my name is Janne Kananen, and I want to thank you for this opportunity to give this talk. I have nothing to declare, but additionally I want to emphasize the role of MD Anssi Koivula in this work, because he was the main man behind these analyses. So, we have previously shown that in Alzheimer's disease, the cerebrovascular pulsations have increased in speed and variance, and these changes are thought to be associated with impaired protein waste clearance. And the understanding of these changes provides insight into AD brain physiology and pathology. In this work, we hypothesized that could the relative harmonic power be used as a non-invasive way to estimate the brain pulsations. So, we wanted to develop a novel approach to leverage ultra-fast fMRI sequences to capture physiological information beyond capabilities of usual conventional fMRI techniques. We made a non-invasive way to estimate MREC waveform complexity by using frequency domain relative harmonic power method to analyze the variability of diastolic effects, such as aortic notch on brain pulsations. The ultra-fast fMRI sequence that we used is called magnetic resonance encephalography that captures high-resolution cerebrovascular impulses up to 5 Hz. The total harmonic distortion is widely used engineering method that is used to assess the relative harmonic power of the harmonic pulsations. The AD characteristic neurodegeneration has been linked to aggregation of abnormally folded proteins in the brain parenchyma, and the MREC has enabled to detect the detection of these brain pulsations. We used Siemens 3 Tesla scanner and with the MREC and TR of 100 milliseconds. So the MREC allows the detection up to 4 harmonics of cardiac cycle, and we calculated the power of wideband harmonics, normalization, narrowband, principal cardiac power. We scanned 34 AD patients and 29 controls, and based on these results, it was seen that cerebrovascular waveform complexity is significantly decreased in AD. The differences were located in brain parenchyma, however, not in major blood vessels or the ventricles. As a conclusion, this study introduced the relative harmonic power metric, which is a novel, fast, frequency-based approach to assess cerebrovascular pulsations. And these findings reveal that altered impulse patterns happens in regions usually affected by AD. These offer potential biomarkers for understanding AD impact on brain hydrodynamics, and as a result indicate the AD brains exhibit more monotonous cerebrovascular pulsations that might suggest reduced responsiveness to neurovascular and physiological demands of brain tissue. And these happen in areas already shown in increased pulse propagation to perivascular conduit narrowing. We want to thank all domestic and international collaborators. Thank you.
Thank you so much. And with that, I would like to invite Manuela Carriero, exploring brain microstructure energy relationships with diffusion MRI and calibrated fMRI measurements.
Okay, thank you for this invitation. I am really glad to be here. So I am Manuela, and I need to declare that my PhD studentship is part funded by Siemens. So the title of this work is Exploring Brain Energy Microstructure Relationships with Calibrated fMRI and Diffusion MRI. Indeed, the aim of this work is to study the relationship between brain energy consumption and the microstructure in order to have a better understanding of the energy efficiency and inefficiency in relation to the structure. This is done using dimensional magnetic resonance imaging techniques and through a data-driven approach. If you try to search in literature about this work, about this kind of relationship, you will not find many works. There are some hypotheses that are based on invasive methods or that use ex vivo data, or there are some hypotheses that have never been tested. In our case, we used, as methods, we used calibrated fMRI, which allows for the estimation of a parameter that is called cerebral metabolic rate of oxygen consumption, that is CMR of 2. This can be considered as a proxy of brain energy consumption. This technique is based on a synchronous acquisition of ASL and BOD images, and this allows to estimate a parameter that is called the oxygen extraction fraction, and through the Fick principle you can estimate the CMR of 2. While instead the microstructural parameters are estimated through the diffusion weighted images, through the estimation of the parameters of a model that is called SANDI. SANDI model has the peculiarity that considers the intracellular signal as coming from two compartments, one that is the neurate and one that is the soma, that is the body of the cells. And so you can have information about the soma radius and the soma volume fraction. These are characteristics that are important in gray matter. In fact, our study has been focused on gray matter. So we acquired data from 39 healthy subjects from a scanner, free Tesla scanner, Siemens scanner. And the tables summarize the characteristics of the sequences that we used. We used for the fMRI calibrated fMRI a sequence that is called the DEXI, that allows for a double excitation, and so for the synchronous acquisition of the ESL and BOLD images. So now I show you some of the results that we obtained. We made two kinds, so in this case, these are the results that consider the relationship between CMR2 and Sum volume fraction. We made two kinds of correlation analysis. One that is the correlation across regions, that is shown through the scatter plot. And the other one is the correlation across subjects, that is shown through the colored map. And you can see that we find, as it is intuitive to think, a positive correlation between CMR2 and the Sum volume fraction. And this positive correlation is also kept across subjects, so in the colored map that you see. This is instead the same analysis performed between CMR2 and Soma sites. This is the most interesting result, that is the CMR2 is positively correlated with the Soma sites. And very interesting are also the colored correlation maps, because you can see that the left side of the brain is characterized by a higher absolute value of correlation. And this is interesting because all the subjects were right-handed subjects. So, in conclusion, further investigations are required to understand the reasons for the associations between CMR2 and Soma structure characteristics. For example, the positive correlation between CMR2 and Soma sites could be due to different firing rates in cells of different sizes, or also different packaging densities. And so the importance of this work is that a deeper understanding of the energy microstructure relationships in the human brain will enable us to characterize efficient brain function in the normal brain and potentially inefficient brain function in neurological disease. Thank you. I want to thank who founded my work, and also my professors and my colleague. Thank you.
Thank you so much. And then we're coming to Sean Warrington with On Harmony, a multi-site, multimodal traveling heads resource for brain MRI harmonization.
Thank you for the introduction. I have nothing to declare. So we understand by now that we need harmonization tools when we're performing multi-site studies, as we know that scanner variabilities and hardware differences can induce or mask apparent biological effects. But what's been lacking so far are objective ways to assess the efficacy of harmonization tools. So we built On Harmony. It consists of 20 participants, eventually scanned on up to 8 3T scanners across 3 major vendors, GE, Siemens, and Philips. 5 modalities, T1, T2, SWI, resting state, and diffusion. And we include within scanner, within subject repeat scans. It's the most comprehensive resource of this kind that we know of. Further, it's also being extended currently to include scanners from the UK Biobank. So it's now openly released, except for the UK Biobank data, as it's not available yet, on OpenNeuro, so you can download it and use it for testing any tools that you like. It consists of 3 phases. Phase A and B both contain 10 participants. Phase A, unfortunately, was interrupted by COVID-19, so it has a slightly longer between scanner intervals. But Phase B has much shorter between scanner intervals, more balanced demographics, and a better split between scanners. Phase C is our extension to the UK Biobank. So far, we've scanned 11 participants on the primary scanning facility in Stockport in the UK. And so far, 10 participants have completed within scanner repeat scans. And they're all on different scanners. So OnHarmony consists of over 200 sessions so far. And as I said, T1, T2, diffusion, resonance state, SWI. Protocols are adapted from the UK Biobank imaging protocol as a starting point, but we respect site practices, scanner hardware limitations, and we try and keep the acquisition protocol below an hour. And you can see a snapshot of the data from a single subject here, and you can see that overall, the image quality is pretty consistent across scanners. So data undergo comprehensive quality checks and processing. The raw DICOMs are converted to NIFTY, and then we do checks for artifacts and any issues in the data. They're then converted to BIDS format before being processed with the UK Biobank pipeline. This gives us thousands of multimodal IDPs, or image-derived phenotypes, and we also extract additional IQMs using MRIQC and EDQC for diffusion. Defaced and anonymized data are then released on OpenEuro. So after basic visual checks, we do some quantitative QC, and basically we're looking at the variability of image quality metrics across scanners and across subjects in our two independent phases. And the takeaway message here is that 80% of our IQMs, the variability is below 1 standard deviation of the mean, and our maximum variability is 2.1, so there are no extreme outliers in our data. Next, we look at the variability of brain features, IDPs, and we can see heat maps here, which are the summary of between session similarities. And as you'd expect, the within scanner repeat scans are much more similar compared to the between scanners. This is something that we would expect. But what we can do is we can start to pool these sources of variability or this similarity into 3 different pools of variability, looking at the within scanner, within subject repeat on the blue, and then the between scanner within subject, i.e. the scanner effect, and then the biological between subject variability. And we see that scanner variability and subject variability overlap quite a lot, meaning, again, that the scanner variability can mask biological effects. Next, we show how we can use OnHarmony to explore different harmonization approaches. So first, explicit harmonization as an example with combat. We apply combat to T1, and as an example, FA from diffusion MRI. And we can use our within scanner variability as a baseline against which we can compare the between scanner variability before and after harmonization. And you can see when we apply combat, our variability does go down, but it never reaches this within scanner variability. This is pretty consistent across the IDPs that we checked. We can also look at implicit harmonization. So this is the optimization of image processing pipelines or tools to be robust against scanner effects. So, for example, cortical segmentation. Here we do a couple of different methods of segmentation, including image-based registration, atlas-based registration method, FreeSurfer, and deep learning implementation of FreeSurfer, FastSurfer. And again, we can look at the within scanner variability and the between scanner variability, and we can make informed decisions about what pipelines we want to use for our data. So these are just 2 examples of how we can use OnHarmony to optimize processing pipelines, develop new harmonization tools. All of the data is available, and the UK Biobank data will be released soon. So if you have some fMRI pipelines and tools that you want to test the generalizability across these vendors, please download it, and you can find out all the details in our 2 papers. Thank you.
Thank you all so much. And now's the moment where we can all vote for our favorite presentation. So please take your phones out. You can either go to the homepage that you see on the top or scan the QR code. I hope it also works for the people in the back. I see some thumbs. Okay, perfect, awesome. I'll give everyone like 30 seconds to scan, and then 40 seconds to scan. In the meanwhile, Patricia can already come up. Everyone okay? Did it work? No.
Okay, so while the voting is being processed, I'll tell you a little bit about the planned future events for our study group. So we have already thought about doing 2 virtual meetings this year. One doesn't have a date, an exact date yet, but we're aiming for July this year. And we'll be using our own virtual platform, the Zoom platform. And the topic that we have selected is neonatal and infant fMRI, a topic that we think is really getting a lot of interesting work being done and very exciting. So we hope we'll find it of interest. The other one will be in November, on the 25th of November. We have already booked this slot with ISMRM, so this will be through the ISMRM platform, and the topic will be preclinical fMRI and also multimodal, which is something that preclinical studies can do a lot, and we also hope this will be agreeable to a lot of the community working with preclinical and also the others who really need to learn about it. We thought of, we discussed a lot of possible topics, and we list some of them here. Open science and reproducibility, physiological fMRI, lots of different topics here, also in overlap with some other study groups that maybe we can do this together with. Cartesian versus the rest of the world, turning up our MR scanners for the different vendors. If there are any suggestions by anyone here, we are really very welcoming any suggestions from you, so very open. So if there's anyone actually keen to organize one of these, because now we can do it without having to go through ISMRM and booking a slot within their very specific slots. We have a lot of flexibility, so we can just come up with a topic, find a date, and do it. So feel free to reach out if you have any ideas. Okay, so because we don't have so much time, another thing that we have already started to think, although this will not be this year, so looking ahead, we really enjoyed the workshop this year. I know some of you could not be there, but it was really fun, and we want to do another one, and we are aiming for 2027. We still don't know where, of course, all of this is still brainstorming, but we think we should be going to Europe or maybe Asia, since the one this year was here in the US. The dates is something we'd like maybe some feedback, so either we could do it just like this year, before ISMRM in March or April, or as we did in Padova a few years ago, early in September or maybe early October. So if people have preferences, again, please do reach out. Maybe not right now, since we don't have so much time. But we are very open to get your feedback. And we also really like the idea of doing this jointly with, at this time, the ultra-high field study group. I think there's clearly overlaps between our group and others. And so we thought maybe we could repeat this. And we consider different study groups, the reproducibility research, which we think really would have a lot to go for, especially since Saskia will be our next chair, and she has a lot of work in this context with Neurodesk. But also perfusion, the perfusion study group obviously has a lot of overlap in the physiological fMRI side of things, and neurofluids, the new kid on the block, which is really, I think, grabbing a lot of our attention. So again, if you have ideas, please reach out. Okay, so I am also just bringing you here some announcements actually from Renzo that of related events that you might not know about and you might be interested in. In July this year, there'll be the 70th Translation Alliance in North America meeting with a lot of fMRI as well as other things in Texas. Later in October, there will be the Minnesota workshop on ultra-high field imaging, and I guess Renzo will be organizing a layer fMRI hands-on workshop. So for those of you, right? I don't know if you want to say anything else about it. So yeah, just to make sure that we can hear your voices, and I think there's at least one. If there's any other business, any other topic that you'd like to bring up, please come up to, I don't know if there's a microphone, but I think we can all hear you. Yeah, Witzke, if you have some. Or... Thank you so much, Witzke. Really important to make sure our study group is live in ISMRM. So I think the voting is complete. So with this, I'll say goodbye, see you in a bit, and I'll pass on to Tony to announce the winners.
Okay, we have a congratulations slide. Yeah. Okay, I'm going last place to first place to make it more exciting. So in fourth place, we have Manuela Carriero. So please come up and collect your... Then we have in third place, Janne Kananen. And then drum roll in second place, we have Sean Warrington. And then we all know what that means. First place goes to Nadira del Rosario. And I think that concludes the meeting. Correct? Cool. That concludes the meeting. Thank you all for joining. I would like to invite the trainees to the front to take a photo together and grab a coffee on your way out because we already paid for it. So.